So now we'll move to RNA modification and diseases, right? Okay. So you remember these modifications? What are the major modifications? M6A or 2-oxymethyl, pseudouridine, mRNA cap, okay, and A2I. So those are the major modifications in any of the RNA and majorly with the mRNA, which is our protein coding RNA, right? And we have discussed writers, eraser, reader, and these readers give you the effect. So that effect could be on splicing, could be on mRNA stability, mRNA structure, mRNA translation, okay. And at the end of the day, if the gene expression changes or alters in a physiological situation, that can enhance some gene expression or stop some gene expression, that can lead to diseases, okay. So these are the modifications known till date, and you see the red ones, okay, those are disease related, and the ones with this uh, uh, a rectangle, so those are the one which can be detected with the sequencing. You see how many modifications are actually there which we cannot do any experiment with. So you don't have antibody, you don't have sequencing technology to detect them, but they are there. So tons of modification are there which need to be explored, and few of them are like related to disease, like someone found, okay, this modification, if this modification is there, so that leads to this disease, or this modification is more or less whatever way it is. So only few are actually known to be like implicated in some of the disease, but most of them are not known. Most of them, like, people have not experimented them, okay. You guys can see, or you should move here. दिख रहा है ना? Okay. So as I said, the modified oligonucleotides, so those can be of importance for therapeutic implications. And if you know some modification is increasing the stability of the RNA, so you can use that for mRNA vaccines, right? So those RNA need to be stable enough for the gene therapy or to expressing the protein in vivo. So those modifications, if we know them well, like which modification is doing what, we can use them for therapeutic applications. And if you know some modification is causing disease, you can reverse that. Suppose you say M6A is causing cancer in general, so that's not true, but I'm saying yeah, as an example. So if you express the erasers and erase the M6A, you might reverse the effect. So those are the applications, how we can use these modifiers or erasers for our use in the disease conditions. Okay, so from starting from the transcription, like we have discussed, like once the RNA is transcribed, the cap is the first modification. Then you have splicing, you have polyethyling, you have other modifications that lead to export of RNA to the cytoplasm, and these M6A and pseudouridine. So these modifications affect the stability and translation of the mRNAs. So all these modifications either regulate the expression from DNA to splicing, or to making the protein. So all these steps are regulated by the modifications. So we have discussed this, right? So one of the most heavily modified RNA is tRNA. So you can see how many nucleotides are actually modified here, and you see the modification near the anticodon region, right? So, and these modifications are regulating the translation initiation or elongation. So they are important for translation. So if you change the modification of the tRNA, you actually change the overall protein synthesis process. Okay. And these are the databases. Like I said, the most important database is this MOD omics. So that is listing around 300 modifications, and there are other databases which enlist the modification and their relation with any kind of diseases. Okay. And we also talked about the detection techniques. So if we are going to talk about therapeutics or use of modification in diseases, suppose you want to see if some modifications are more prevalent or less prevalent in any of the disease, you say cancer, diabetes, anything, how will you find that out? By sequencing, right? So antibody based sequencing, chemical modification based sequencing, or your direct sequencing through nanopore. And these are the modifications you can do by NGS, and these are by the nanopore. So but there are hundreds of more modifications are there for which we don't have a technique actually to detect them in any of the disease conditions. So this is antibody based RIP-seq, antibodies like M6A pull down and then look at which fragment or RNA you have more of the modification. This is chemical based, like you stop the reverse transcriptase, right? Or you incorporate a mutation. That way you can, in the sequencing, you can read the mutation and then say that nucleotide is actually modified. It's not a regular nucleotide in the RNA sequence. And this is the nanopore, right? So you have the RNA, it can go for direct nanopore sequencing where you add the adapter with the motor protein, and then that goes to the nanopore and then go for sequencing, right? That is what we discussed in the first class. Here we'll see how that is important for disease. So as I said, any change in the RNA stability translation can lead to diseases. If some genes are important for physiological process, any gene you know that is important for development of cancer? P53 कौन सा? MIC, okay. RAS, okay. P53, P51 और हाँ, okay. हाँ. So there are many genes which are known for the mutation, but apart from mutation, even if there is no mutation on the RNA itself, but if they are heavily modified and you are changing the stability or the translation, then also it can cause disease because the protein formation from that RNA will not be normal. So if some modification are inhibiting, like inhibiting translation, or they are inducing the degradation, so RNA will not be there, so you will not have the protein. Even if there is no mutation, you still don't have the functional protein in the cell. So that can lead to diseases. So cancer, neurological disorder, metabolic diseases, and viral infection. So all those are affected with RNA modification, which are known for the dysregulation. And also RNA modification lead to immune cell function, because you are changing the mRNA expression or the protein expression, you are basically changing the activity of the immune cells, and that also affects the immune response. Okay. And in cancer, there are like tons of papers on modification on cancer, and in all kind of cancer actually RNA modification have been found, and those are mostly like M6A and this 5MC. So it's not that other modifications are not there just because you don't have a detection technique or the antibody. So other modifications are not well studied. Maybe when we have a proper technique or antibody for other modification, you will see more papers. But for now, if you search for RNA modification, 95% or 99% papers will tell you about M6 modification, because you have the antibody to study, like do the pull down, do dot blot, and do whatever assay you want to do. For other modification, we don't have the reagents to study them, so you don't see their implications in diseases or any of the cell physiology. And mostly, as I said, all papers you will see M6A, because it is heavily studied because of the antibody, and 2-oxymethyl is also very well studied. And this example I have shown you already. So this M6A modification can regulate cancer. And in cancer, so if you, as I said, like if you change the modification pattern of the mRNA, you can change the expression of the genes. So that can lead to any therapeutic implications in disease. If you know this modification is causing M6A is causing this disease, you can either overexpress your or less. If this metal 3 you can overexpress, and then for the tumor growth reduction, inhibit metal 3, reduce the tumor. So you can, depending on which modification does what, and based on that you can either overexpress the eraser or the induce that or reduce whatever modification is happening. Based on that you can devise a therapeutics for that disease. Lab is interested in is a metabolic disorder that's called diabetes. Everyone knows diabetes? हाँ, डायबिटीज क्या होता है ये सबको पता है. हाँ, इंसुलिन प्रोडक्शन बीटा सेल से नहीं होता है, कम होता है. ओके, क्यों कम होता है? Okay, so autoimmune destruction of beta cells you are saying. So that is basically type 1 diabetes. Okay, and then जो reduction in insulin secretion बोल रहा हो, so that is basically diabetes mellitus type 2. Okay, type 2. So वो कब होता है? नेचुरल इंसुलिन रेजिस्टेंस. इंसुलिन रेजिस्टेंस कहाँ होता है? बीटा सेल में नहीं होता है, जो रिसेप्टर ऑर्गन है. ओके, ग्लूकोज कौन से ऑर्गन को ज़्यादा चाहिए? मसल और लिवर स्टोर होता है ना ग्लूकोज. ओके, अगर इंसुलिन को रेस्पॉन्ड नहीं किया मसल और लिवर, so your all glucose will be in the blood flow, so the glucose level goes up. So that is basically known as diabetes. But the underlying cause is either your glucose resistance, insulin resistance, or you don't have enough insulin produced from beta cells that can induce the glucose uptake. Okay, both conditions can lead to your increase in glucose in the bloodstream. So one of the reasons why your beta cell doesn't produce insulin is cells don't respond to glucose, first thing. Okay, and if you have high glucose for a very long time, okay, matlab for years, then usually what beta cell does is anytime you eat, now we had the biscuit, tea and other things, right? So the glucose goes into the blood, that goes to beta cell and tell the beta cell, okay, now you have more glucose, secrete more insulin, so that muscle and your liver can take up the glucose. Okay, but if you have that high glucose persistently, then the beta cell is getting that signal every time. Okay, so after a long period of time it will stop responding. Okay, so लाइक तुम किसी को बुलाओगे कि चलो आओ चलते हैं, आओ चलते हैं, वो एक बार, दो बार, 5 बार, फिर वो जब रिडंडेंट होता रहेगा फिर बोलोगे अरे चलो छोड़ो जाने दो अब नहीं जाएंगे इसके साथ, है ना? तो beta cell also does the same thing. For a longer period of high glucose or high fat, it will stop responding. It will not produce the insulin anymore, or produce in a lesser amount that is not sufficient for the glucose to be internalized into the other organs, and that leads to diabetes. Okay, so here what we will see is how RNA modifications, basically mostly as I said M6A, always you search for any paper on any disease, you will always see M6A modification and their readers, writers, and their role in the glucose metabolism, insulin resistance, or development of type 2 diabetes. So one is major organ is your liver for diabetes, and the other one is pancreatic beta cells. Beta cells कहाँ होता है? islets of Langerhans. ओके, कितना % बीटा सेल होता है सेल्स पैनक्रियास में? आधा नहीं, और पैनक्रियास और क्या करता है? इट्स नॉट ओनली फॉर इंसुलिन प्रोडक्शन, इट हैज अदर फंक्शंस, राइट? डाइजेस्टिव एंजाइम्स. ओके, डाइजेस्टिव एंजाइम कौन से सेल से आते हैं? लिवर से? नहीं, पैनक्रियास से पूछ रहा हूँ. पैनक्रियास में क्या क्या सेल्स होते हैं? एंडोक्राइन सेल्स होते हैं, एक्सोक्राइन होते हैं, राइट? एक्सोक्राइन सेल्स, ओके, एसिनर सेल्स जो होते हैं, एंड जो एंडोक्राइन है वो है आइलेट्स ऑफ लैंगरहैंस. उसमें अल्फा, बीटा एंड गामा, डेल्टा. ओके, ये सब सेल्स होते हैं. एंड the cell that produce insulin and important for diabetes is beta cell, and the islets are hardly like 1%. Okay, and in any of the islet like 70-80% are beta cells actually. So those beta cells produce insulin, and this insulin production is not like you are only synthesizing insulin. So insulin, how big is insulin, you know? How many amino acids? 52, 55. मुझे भी याद नहीं है, तुम गलत बोलोगे मैं भी नहीं बता पाऊंगा. कितना है मुझे भी याद नहीं है, बट देयर आर 2 chains. Okay, but those 2 chains of amino acids come from one polypeptide actually. It is synthesized as a single chain amino acid and then it is processed. So one peptide is cut out from insulin, the C peptide comes out, then you have 2 peptides linked by the disulfide bond, and that makes the insulin structure basically. So all this processing, like from, it is not a single protein, like a single polypeptide chain. So that processing, like from the single polypeptide chain to 2 polypeptides and giving the insulin structure, needs lot of enzymatic reactions. And then after the insulin protein is actually synthesized, it need to be secreted. okay, and secreted in response to glucose. ऐसा नहीं कि हमेशा secrete होता रहेगा, हमेशा होता रहेगा, but it will be in a smaller amount. But when we have glucose, like we had breakfast now, so now the cell need to secrete more and in a quick response. Like it should not be like taking hours to send the insulin to the blood. So whenever glucose stimulation happens, within minutes you have an insulin burst from the beta cells. After that, the insulin mRNA, after like an hour or two, your transcription goes up. But initially transcription doesn't go up. Initially cell respond, okay, now glucose is there, let me secrete. So that response pathway, secretion, needs many proteins. So those also need to be expressed in a specific amount. And then after the secretion, so now the reserve is no more there, like reserve has depleted. Now beta cell respond to that depleted insulin and synthesize more insulin. So that induces insulin translation in the beta cell. So once that is that translation is done, then you have more insulin for the next cycle. If the glucose is still there, it will still go on secreting more insulin, or you will have as insulin granules. So all this process needs lot many genes, not only a single gene which is responsible like insulin. For the synthesis to secretion, you have a cascade of genes which need to be expressed at a specific level to give you the actual secretion of insulin and maintain the body glucose level. So in case of diabetes, okay, type 2 diabetes, you can see the modifiers, readers and erasers, they also alter. So because of this alteration, the gene expression changes. And here, like pancreatic beta cells, you see M6A goes down. So if you think, okay, how can I reverse that? What you will do? Either block eraser or overexpress reader, the writers. Okay, so that you can reverse it back. But you need to know how much you need to write, so that you like exactly bring the expression level to the basal level. ऐसा नहीं कि फिर राइटर्स हर जगह M6 कर दिया 100-fold over express करके. So that comes where the precision therapeutics comes. Like now whatever we are, like for all the diseases we are getting treated is with the chemicals, like the compounds, and those are general chemicals. For some people they work, for some people they don't work, okay, based on the physiological uh, state of the person or the mutations that person has, or the food habit, the physiology, everything comes into play. And for the same thing, like if you want to change the modification, you need to know what kind of modification, which genes are modified, and how much you need to modulate that. Based on that, you can have a therapeutic application. And this is also diabetic cardiomyopathy, where M6A goes down. Okay, here in this is type 1, this is type 2, this is goes up, and your FTO goes down. So if you want to reverse, you have to do the reverse basically. Okay, you can overexpress FTO and then you can get back the normal physiology. So this is one way how you can use RNA modification to reverse any disease, but this is not in pipeline. Okay, not in, like this is still under research. Like it is like so many modifications are there, and because one eraser or one writer is changing multiple genes at a time. It is not that only one gene, like I showed, like one gene is getting regulated. You have thousands of genes which have M6 can be modified. So you are basically changing all the genes when you are overexpressing or reducing any of the reader or writers. Okay, so there are lot many modifications have been known for different genes on different RNA in different diabetic cardiomyopathy or diabetic nephropathy. So different conditions have different modifications. We'll not go into the details, but the bottom line is RNA modification can cause disease, and if you can reverse that back by knowing which modification is doing what, you can have a therapeutic application. Okay. And as I said, like mRNA vaccine, so if you have modifications that can stabilize the RNA, so you can have mRNAs, those are stable, right? So but in the first class, as I said, my lab works on circular RNA, which are not modified. Some are modified with M6A, but without even any modification, they are very stable because they do not have 5' or 3' ends. So they can act as a therapeutic, like can be used for therapeutic purpose in the gene therapy. And in the future, as I said, only few we know by sequencing, or we have methods for their detection, and the others need to be discovered, and how they are playing a role also need to be studied. But we need first detection methods, antibodies, or any pull down method to study how these modifications are affecting different diseases, right? Yeah, so better methods for detection, better method for bioinformatic tools for identifying, and then therapeutic application. So lot many things need to be done in this area, like the RNA modification. It's not a very well studied because it is very dynamic. Like I said, the DNA modification, like you only have 2-3 modifications and others are on the protein, but here you have hundreds of modifications and they are dynamic. So you need lot more effort to study them and find out how they are regulating a specific disease and how we can have a therapeutic application using those modifier, so erasers or the readers. Okay, any questions? No. We'll go back to diabetes. So that my one of the focus area of my lab, not exactly diabetes, but we work on pancreatic beta cells, okay, that secrete insulin. And there are already lot of treatments. Any drug you know for diabetes? Metformin, the most famous one. और पढ़ो मत, बिना पढ़ के बोलो, वो तो लिखा हुआ है बहुत सारा ड्रग. हाँ, सुनाई नहीं दे रहा है. मेटफॉर्मिन, हाँ वही बोला, मेटफॉर्मिन वही कॉमनली यूज होता है विथ कॉम्बिनेशन. ओके, बट डायबिटीज इज क्यूरेबल? नो. क्यों? प्रिवेंटिव है बट क्यूरेबल नहीं है. क्यों नहीं है? ये मत बोलना कि मॉडिफिकेशन से पता नहीं है. हा? वो भी एक रीजन है, क्योंकि हमें सब कुछ नहीं पता है. ओके, few things we know in terms of gene expression or the way some genes are expressed at what level and how, and in response to which environmental factor or our physiology. See, so all that play a major role. So most of the time we think okay mutation है तो कुछ disease होगा, ऐसा नहीं है. It's not always कि one mutation is. There are mutations which are like dominant mutations, like you have a truncated gene, you do not have the protein, that can cause a disease. But there are many other mutations which need additional factors to have the disease. Like पहले diabetes इतना नहीं होता था नहीं होता, अभी क्यों हो रहा है? Lifestyle. It's not mutation के over 20-30 years we have accumulated many mutation. No, ऐसा नहीं होता है, ठीक है, but it is because of the lifestyle, the way. Epigenomic, that is little hard to say, but जो mutations है वो तो next generation को जाएगा. So some mutations are inherited and that cause disease, diabetes, but not all. Okay, and mostly it is lifestyle. Okay, and as I said, like you have high glucose or high lipid for a very long period of time, that will induce in the first, like in the short term, you will have more beta cells, more insulin secretion, and then when the glucose level goes down, beta cells also think, okay, everything is normal now, I can rest. Okay, but if it happens for a very long time, so then the beta cell will go into apoptosis, because every cell has a lifetime and how much they can work. Okay, so you cannot like be running every day like for 12 hours. You can go for exercise for half an hour and then rest, then do work, then maybe in the evening you can go to gym, but you cannot run all the time. So if you make the beta cells run continuously for long time, they will be sick, they will die, they will go into apoptosis. So you will not have enough beta cells to produce insulin, and that will lead to diabetes, right? And my lab is looking at like what happens when you treat the cells with glucose in terms of changes happening in the long non-coding RNA, specifically circular RNA, how it is getting regulated, and how this circular RNA regulate the gene expression in beta cell that is important for beta cell growth. Okay, and you all don't know about circular RNA, right? So this is not a new RNA type. It was actually discovered in 1980s in electron microscope, but people thought that could be a artifact. Okay, could be splicing errors or lariats. You know what is lariat, right? What is lariat? Splicing के टाइम पे जो इंट्रॉन का सर्कल निकलता है, so that is basically your lariat, and people thought, okay, this could be lariats or maybe splicing errors, we don't know. For 20 or 30 years people ignored it. There were papers talking about RNA circles, but people thought those could be like just sporadic expression, like one gene giving rise to a circular RNA. Like I said, now we don't have method. Then in 1912 and 13, there were few papers. So they identified RNA circles in all kind of cells. All eukaryotes have thousands of circular RNAs. And from that point, and how they discovered it, they never wanted to find the circular RNA. They wanted to see chromosomal misarrangement, and like the bioinformatic tool used to see any kind of misarranged sequence. So when they were looking at misarranged sequence, people saw misarranged exons, like your exon 2 is coming after exon 3. So that's only possible if there is a jump, that you can say, okay, scrambled exon. When the splicing is happening, somehow by mistake 2 goes after 3. Okay, that is one possibility. The other possibility is if it is a circle, like 2 end of the 3 joins the beginning of the 2. So those are two possibilities. So in the sequencing, the circRNA expression in all kind of cells, and many cells they saw that is a ubiquitously expressed RNA type. Okay, it's not there in the books till now, it's not a very long time, but there are lakhs of circular RNA expressed in different organisms, and this is abundant, conserved, and they show tissue specific expression. And how they come is, like I said, like the end of exon 2 can attack the start of exon 2 and make a circle. The end of exon 3 can join start of exon 2, or sometimes the intron lariat itself. Intron lariat is degraded by what? Which enzyme degrades? Debranching enzyme. Okay, so how the lariat is different? Is it a 3' to 5' joining or something else? It's a 2' to 5' joining. Okay, it's not 3' to 5' joining. So, but there the debranching enzyme can degrade the branch point so that the intron now have the 5' and 3' and the exonuclease can degrade it. Okay, but in case of circRNA, there are elements in the intron that make them stable. They make them resistant to the debranching enzyme, so they are not degraded by the debranching enzyme, and they remain as stable circles. And sometimes, like your exons, they join, like 2 exons are there, the middle intron also get spliced out, like stay with the exon, and you have exon intron circRNA. So from one gene you can have multiple circular RNAs. Okay, so how many genes we have? भूख लग रहा है? जितना जल्दी बोलोगे उतना जल्दी क्लास खत्म करेंगे, फिर लंच के लिए जाएंगे वो. हाँ हाँ, 23,000. चलो, so 20 to 25,000 genes. But when you look at circular RNA, I said like a million circRNA have been found in the sequencing. So that means like multiple circular RNA are coming from a single gene. Not all genes make circRNA, but many genes, they give rise to multiple circular RNA. So that gives rise to the number. Then how we detect them? Suppose exon 2 and 3 are joining. Okay, what is the easiest way to detect? Gel. हाँ, जेल पे कर सकते हैं. So how that will move? Suppose you have a 100 base pair RNA circle, 100 base pair linear one. Circle will be moving faster. Okay. Okay, so 2D gel electrophoresis you can do. So, and also another thing you can do is the trap. Suppose when something is a circle, so you are putting it in the agarose. Agarose solidifies, okay, and then that traps the circle. But the linear will never be trapped, right? You can put the electricity, linear will flow in the pores, but circle is already like trapped in the agarose polymerization, and then they will stay there. So that is another way to separate them from linear. What else you can do? Easier ones कोई बता नहीं रहा है. सर, PCR, right? So if you know a gene and you want to see if the gene is circle or linear, you can do PCR. सबको PCR पता है? हाँ, PCR में क्या क्या होता है? क्या चाहिए PCR के लिए? पॉलीमरेज, टैग पॉलीमरेज, प्राइमर. कितना प्राइमर? फॉरवर्ड एंड रिवर्स. ओके, तो अगर कोई जीन सर्कुलर है उसको कैसे प्राइमर डिजाइन करोगे? एक प्राइमर, हाँ बताओ बताओ। आरएनए है, आरएनए है, रेस्ट्रिक्शन एंडोन्यूक्लियस डोंट वर्क ऑन आरएनए। हाँ प्राइमर, एक प्राइमर किसने बोला? ओके, एक प्राइमर से कैसे होगा? वो तो ऐसे घूमता रहेगा। यूनिट